Good morning, student. Today we are going to see protein synthesis, that is also called as gene expression. Genes is a segment of DNA that can synthesize at least one protein. DNA expresses itself through protein synthesis. Genes, that is DNA, cannot express it directly. It always expressed by the formation of protein. The protein we eat goes into our intestine, where it gets broken down into amino acids. The amino acids are shifted to each and every cells of our body, that is living cells of our body, where it is again utilized as per the need of our body, that is the genetic code present in the DNA. So DNA expresses through mRNA. Process is called as transcription. Protein synthesis involves two processes. One is transcription and other is called as translation. So during transcription, the DNA produces mRNA on one of its strand called as template strand. DNA अपने एक स्ट्रैंड पर mRNA प्रोड्यूस करता है with the help of RNA polymerase. RNA polymerase bind to the primer. Primers are the 17 to 30 nucleotide long piece of RNA nucleotides. RNA primer is formed with the help of RNA primase. Here we can see the transcription process, which involve formation of mRNA on DNA strand, is called as transcription. The DNA contain genetic code in the form of codogen, which is nothing but the triplet of nucleotides, that is A, T, G, C, A, T, G and C, and its different arrangement produces different types of codogen on DNA. It transcribes into mRNA with the help of RNA polymerase. RNA polymerase binds at 3' end of DNA and slide past towards the 5' end. RNA formation occurs with the help of transcription factor, that is sigma factor and rho factors are involved in the formation of mRNA. Sigma factors are the starting factor and rho factors are the terminating factors. Sigma factor binds at the 3' end of DNA and rho factor binds at the 5' end of DNA. Due to sigma factor, the transcription process begins. RNA polymerase binds to the DNA strand through RNA primer. It slides past and produces new strand called as mRNA. When it moves from this point to this point, it goes on adding new nucleotides of RNA. When the transcription process is over, the RNA comes out from this loop. It is useless mRNA. This RNA is useless. That means it is of no use. It is non-functional because it contains some non-coding region in eukaryotes. These non-coding regions are called as useless or intron regions. Introns should be eliminated with the help of a process called as processing of mRNA. During processing, three main steps occur. One is called as capping, splicing, that is deletion of this intron, and lastly polyadenylation, that is formation of poly A tail. Capping occurs at 5' end, while at 3' end many adenine nucleotides goes on adding, that is called as polyadenylation. It is also called as poly A tail. The mRNA now form is a functional mRNA. It is now ready for translation. The translation occurs in cytoplasm. The mRNA formed inside the nucleus of eukaryote. For translation, it has to be comes out from nuclear pore into the cytoplasm, where ribosomes are scattered. That means large subunit and small subunit of ribosomes are scattered in the cytoplasm. With the help of magnesium ion, the two units comes closer to the mRNA and bind to the mRNA. Firstly, the small subunit binds to mRNA. Then second step occur, that is a binding of tRNA amino acid complex that brings first amino acid to the start codon. Start codon is called as AUG. Adenine, uracil and guanine sequence on mRNA is called as start codon. Codons are the sequence of 3 nucleotides present on mRNA, while the 3 nucleotides present on DNA constitute codogen. Both are the codes language. Now, the large subunit binds to the ribosome with the help of binding factors, that is magnesium ion. Now, this step is called as initiation step. This step is called as initiation step. After initiation, elongation occurs, or you can say polymerization occurs due to addition of amino acid to the ribosome. One by one, tRNA amino acid complex enter through A site and then moves to the P site. After adding phosphodiester bond, after the formation of phosphodiester bond by between the two amino acid, that is previous and new one, the ribosome is shifted one codon ahead. Finally, the ribosome reaches to the stop codon, that is terminating point. In stop codon, in termination step, what happen? All the constituent get separated, that is large subunit, small subunit, then large subunit, small subunit of ribosome, then mRNA, polynucleotide chain, all get separated, and empty tRNA also. This step is called as termination. Now the amino acid is ready for processing. After processing of polypeptide, disulfide bonds are formed and polypeptide is now converted to protein, which is then converted finally to either enzyme or structural protein. In this way, DNA expresses itself to form protein. So DNA always express through protein synthesis. Let us see the example. Our skin color is due to protein, melanin. Our skin color is due to protein called as melanin. So melanin is synthesized. Melanin is a protein which is synthesized from DNA through mRNA and ribosome. Let us see another example, that is growth hormone. Growth hormone is also a protein, peptide hormone. That means it is a protein. So protein is formed only through this process, not any other process. So another example is insulin. Insulin is also a protein which is formed through transcription and translation. In this way, DNA expresses itself. That means it shows the characters, it develops the character through protein synthesis only. Thank you. Next time we will see about genetic code. Thank you.